Hello students. So, welcome. I would like to welcome you all to the first tutorial session for this NPTEL course, um, Introduction to Cell Biology. And let me introduce myself. I am Dhruba Chatterjee, a PhD scholar at the Department of Biological Sciences at IISER Mohali. So, first, I will try to run you through a summary of whatever has been taught in the first week of this NPTEL course, Introduction to Cell Biology. I hope that you have already gone through the lectures of the first week, and then I will also try to address some of the questions that you have been asking in the discussion forum, and then um, I will also try to run you through and solve together kind of some of the questions that had been asked in the previous assignments of this course, so that it can be maybe of some help for you in solving the questions that had been asked in the current week's assignment. Okay, so let's start. Um, okay, so in the first week of this course, Introduction to Cell Biology, the main focus was on the molecular biology. And the primary one important concept that was being discussed for quite some time is the central dogma. So, as you already know by now, that DNA, that is deoxyribonucleic acid, is a primary genetic material and the biologically important genetic information in terms of which is actually stored within the deoxyribonucleic acid, that is DNA, in terms of the nitrogenous base sequence, that is nucleotide sequence. Now, DNA is capable of replicating itself. That is, that the DNA, the parent DNA strand can act as a template and give rise to new DNA strands with the help of enzymes which are called DNA-dependent DNA polymerase. Now, DNA is also transcribed using DNA-dependent RNA polymerases into RNA, that is ribonucleic acid. There are various different types of RNAs it can be. It can be mRNA, tRNA, rRNA, as well as various other different types of RNAs. Now, among all of these RNAs, for example, mRNA is the RNA which is synthesized within the nucleus by these DNA-dependent RNA polymerases. But after that, the RNA comes out of the nucleus to the cytoplasm, where it is translated into where the nucleotide sequences are being read by the ribosomes, and the nucleotide sequence is being translated into proteins, where basically these amino acid tRNAs bring these different amino acids, and the amino acids are joined together by covalent bonds in a certain sequence to form the proteins. Now, RNA can also act as a template. RNA can also act as a template for the replication and formation of more RNA molecules using the RNA-dependent RNA polymerase. And this is especially important for, this is especially seen in case of, for example, viruses, RNA viruses, for which the RNA is the primary genetic material. And after these viruses infect the host cells, there the viral RNAs are being replicated using the viral RNA-dependent RNA polymerases. Yeah. And there is a class of viruses which are called the retroviruses, an example of which is the HIV, human immunodeficiency virus. So, uh, what in them, what happens is after these viruses infect the host cells, within the host cells from the viral RNA using the viral reverse transcriptase. That is RNA-dependent DNA polymerase. What happens is a complementary DNA strand is actually formed from the RNA. So this gives you the primary flow of information, the flow of genetic information of central dogma, that the DNA or deoxyribonucleic acid is the primary genetic information, is a primary repository, is a primary molecule which serves as the genetic material, is the molecule in which the biologically important, biologically necessary genetic information is stored. Now, the DNA is capable of replicating itself using DNA-dependent RNA polymerase. From DNA, the RNA transcripts are made, mRNA transcripts are made, or the different RNAs are made by transcription using RNA-dependent RNA polymerase. The RNA information, especially the mRNA information, is read by the ribosomes and translated into proteins. RNA can also act as genetic material for in some cases, for example, in case of some RNA viruses. And for example, after these viruses, these RNA viruses infect their host eukaryotic cells, there the RNA is actually replicated using RNA-dependent RNA polymerases. And in case of retroviruses, from this RNA, the DNA is formed using RNA-dependent DNA polymerases, which is basically the reverse transcriptase, right? Okay. So now the next aspect that we will try to focus on is that what is the chemical nature of this. So DNA is the genetic material. DNA is the biomacromolecule in which the biologically important genetic information is stored. What is the chemical nature of this DNA, deoxyribonucleic acid? What is DNA made up of? As well as what is the structure, the three-dimensional structure of DNA, right? So so deoxyribonucleic acid is actually a polymer. It's a polymer of small units which are called the DNTPs, that is deoxyribonucleoside triphosphate. Now you already know some basic things. For example, the DNA is double helical. It's an antiparallel double helix, and these two strands of the DNA, two DNA strands, are held together because of Watson-Crick complementary base pairing between the nitrogenous bases, between the purines and pyrimidines. So between the purine adenine and pyrimidine thymine via two non-covalent hydrogen bonds, and between the purine guanine and pyrimidine cytosine via three non-covalent hydrogen bonds, right? So let's look into a bit more details of the components, the small units which polymerize to form this deoxyribonucleic acid, that is DNA. So DNA, as I already mentioned, so is a polymer of these DNTPs, that is deoxyribonucleoside triphosphate. Deoxyribonucleoside triphosphates, right? So these DNTPs actually consist of three components. One is the sugar, the deoxyribose, the 2-deoxyribose pentose sugar monosaccharide. Okay, pentose because it consists of 5 carbon atoms, and 2-deoxyribose because, so for example, ribose is a pentose sugar. It consists of 5 carbon atoms, right? But DNA, so it consists of 2-deoxyribose. You can see that as a 2' carbon instead of OH, there's a deoxylation and it has just hydrogen. And that is also very important because in case of RNA, what happens is they consist of ribonucleoside triphosphates. There the sugar backbone is ribose, that is 2' OH, 2' carbon OH. OH attached to the 2' carbon. Whereas in DNA, you have 2-deoxyribose, where at 2' carbon, you have hydrogen. There's a deoxylation. That is important because this OH group actually acts as a very important nucleophile, which allows RNA some very important catalytic activities, and there are RNA enzymes, which are called ribozymes. But this reduces the stability of RNA. It is much less inert. It has catalytic capabilities, but it is much less stable. Whereas DNA, because of this 2-deoxyribose, is much more inert, much more stable, but however does not have catalytic activities. But because of this increased stability, DNA is chosen to be the genetic material mostly in the eukaryotic cells. In the eukaryotic cells, DNA is the genetic material. Whereas RNA can act as transcripts, which have shorter half-lives. They are synthesized, they do their purpose, and then they are degraded. And also RNA in many cases has the catalytic activities, whereas DNA is chosen as genetic material in eukaryotic cells. Whereas, however, RNA also acts as genetic material in some cases, for example, in the case of the viruses, the RNA viruses. Yeah, so now let's come back to this DNTP. So this deoxyribonucleoside triphosphate, it consists of this deoxyribose sugar, right? And to the first C, to the C1, is attached to nitrogenous base, which can be purines, that is adenine or guanine, and pyrimidine, that is thymine and cytosine. And to the 5' carbon is attached to your phosphate, is attached to your triphosphate actually. So that makes your DNTP, that is DNTP or nucleotide, deoxyribonucleoside triphosphate. Until this triphosphate is attached, it's called a nucleoside. When this triphosphate is attached, it's called nucleotide. And these nucleotides, that is DNTPs, that is sugar associated with this nitrogenous base at C1 and associated with the triphosphate at C5, gives you the smallest unit that can polymerize to form DNA. Now, how do these? Now, let's look into the chemistry, chemistry of how do these DNTPs polymerize to form the polymer, that is the deoxyribonucleic acid. So, if you look into the chemistry and how the chemistry is, I will tell you first, and then we will also look into the figures. But the 3' OH in each of these DNTP, you can see the 3' OH is free. So what happens is the 3' OH, the free 3' OH of one DNTP can actually act as a nucleophile and attack the phosphate, this phosphate of other DNTP. Okay, so what happens is, and that causes this PPI to come out as a living group and leads to the formation of a new phosphoester bond, between this DNTP and the incoming next DNTP. Okay, so you can see here as well that here is a schematic where you can see this. So this is already a DNA strand. This is already a DNA strand with, for example, in this case, it has 3 DNTPs which have been linked together, which have been polymerized together using this phosphodiester bond. The phosphodiester bond, which is formed between the 3' carbon of one DNTP and the 5' carbon of the next DNTP, right? So how has that been formed? That you can see while a new DNTP is getting polymerized. So when a new DNTP is to be polymerized, it can only be added to the 3' end, because only at the 3' end you have the free 3' hydroxyl group, which can act as the nucleophile. This nucleophile attacks this phosphate, which is attached to the 5' carbon of the next incoming DNTP. Now that what it causes is it leads to the formation of a new ester bond, a new phosphoester bond here, and the PPI, this diphosphate, actually comes out as a living group. It actually comes out as a living group, and you get a new phosphoester bond here, and that leads to the formation of a new phosphodiester bond, then brings together these two consecutive DNTP. Right. So now you can appreciate that when this new DNTP again comes, then it's the 3' OH of this new incoming DNTP is again free and is capable of attacking the phosphate of another DNTP, the phosphate attached to the the 5' carbon of another new DNTP which is coming in, and carry on the polymerization. So you can understand that DNA. Now you can, I think you can appreciate this fact that so DNA has a directionality in this sense that on one end you have the 5' phosphate. The on one end you can see that the 3' carbon is the hydroxyl group of the 3' carbon is involved in the phosphodiester bond, whereas the 5' carbon is not involved in the phosphodiester bond. It has instead the triphosphate. So this end will be called the 5' end, right? Whereas on the other end, you can see that the 3' OH is actually free. The 5' carbons, 5' carbons phosphate is involved in the phosphodiester bond, and the 3' OH is free. So this end will be called the 3'. Now, since at the 3' end, only at the 3' end, you have this free hydroxyl group at the 3' carbon atom, this which can act as a nucleophile and lead to the polymerization of a new DNTP that is coming in. So new DNTPs can only be polymerized at the 3' end. That is, DNA always elongates from 5' to 3' direction. DNA always elongates from 5' to 3' direction because new DNTPs can only be polymerized at the 3' end, which has the 3' hydroxyl group and can act as a nucleophile, which is necessary for formation of the new phosphodiester bond. So DNA polymerases, which will also, with this information and concept will be handy when we will be trying to understand the DNA replication. Because there we will see that the DNA polymerases only extend or elongate DNA in the 5' to 3' direction, right? Now, as we have already um, seen in the lectures that DNA is a antiparallel double helix. So what that really means is that, for example, in this right side figure, you can appreciate this thing that for this strand on the lower end, this 5' carbon's phosphate is involved in the formation of the phosphodiester bond, whereas the 3' which is free. So this becomes your 3', right? Whereas this top end of the left strand, you can see the 5' phosphate is free, whereas the 3' hydroxyl group is involved in the formation of the phosphodiester bond. So this becomes your 5', right? Whereas on the other strand, you can see that here the 3' hydroxyl group is free. So this becomes your 3' and this becomes your 5' because the 5' phosphate is free on this end, whereas the 3' hydroxyl group is involved in the formation of the phosphodiester bond. So if you look at the directionalities that this DNA strand is, let's draw arrows from 5' to 3' because that is how DNA gets polymerized. So you can see that the two DNA strands runs in opposite directions. That's why they're called antiparallel. But this antiparallel arrangement is also necessary for allowing the hydrogen bonding, the non-covalent hydrogen bonding between the purines and pyrimidines of two different strands, right? Between the Watson-Crick base pairing, uh, the non-covalent hydrogen bonding between the purines and pyrimidines of two different DNA strands, like 2 hydrogen bonding between adenine and thymine, and 3 hydrogen bonding between guanine and cytosine, for ensuring that the DNA is double helix, right? So DNA is an antiparallel double helix, where the two strands run in opposite directions. And this double helix is possible because of the non-covalent hydrogen bonding, the complementary Watson-Crick base pairing between the purines and pyrimidines um, of the two different strands in a specific manner, like adenine pairing with thymine and guanine pairing with cytosine. Okay. Now, I will also try to discuss this slide a bit. So, about what are the different DNA conformations. So, there had been also quite a bit of doubt as I was going to I was also listening the lectures, that what are these DNA conformations. So, I will like to conformation. Conformation is about actually DNA structure, the three-dimensional DNA structure, right? So all of these, so for example, all of these forms of DNA, the bDNA, aDNA, and these two helices are kept together because of um, this, in at least in bDNA, aDNA, and zDNA, these two helices are kept together via non-covalent Watson-Crick hydrogen bonding, that is Watson-Crick base pairing between the purines and pyrimidines of the two different DNA strands. But so how they differ is, they differ in the overall structure of or structure or the three-dimensional orientation of these different of the sugar of the nitrogenous base and various different atoms. So um, that's how they differ in some important characteristics. For example, the BDNA and ADNA are right-handed. Different DNA conformations, and what is the biological significance or relevance, or is there any biological significance or relevance of the different DNA conformations? So these different three-dimensional structures actually differ. So as I said, that they are chemically similar, chemically very similar. There's a three-dimensional orientation of the sugars, nitrogenous bases, and their packing is different. For example, BDNA and ADNA are right-handed, whereas the ZDNA is left-handed. And you can see this some other characteristic features. For example, that what is the distance between two bases, right? So you can see that, for example, BDNA, there are 10.5 bases per helical turn, right? So and this one turn, total one turn is 3.4 nanometer. So the distance between two bases is 3.4 angstrom, that is 0.34 nanometer, very easily obtainable. Whereas this A form is a bit more compact. It's there the distance between two bases is much lesser. It's 2.6 angstrom. And this A form, the bases are also quite a bit tilted as compared to BDNA, which is also represented by this base tilt. For example, if you draw a straight line, then this is the axis of the DNA, right? You draw it on both of these. Now we draw a line which is perpendicular to this axis, right? Like this. So now you see that this is your one base. Sorry. So if I just try to, this is your one nitrogenous base of BDNA, right? So if you see the angle at which it is, it's almost perpendicular to this axis. So it's almost like at 6 degrees. If this is your this line, then your base is just a bit tilted, 6 degrees. Whereas if you see in ADNA, if this is your base, then the angle is much more. The angle is like 20 degrees. So there is a much more tiltedness in the ADN. So that is how that's why they are said to be the DNA different DNA conformations, because the nature, the chemical nature is same. All of these, but the three-dimensional orientation of the sugars and the nitrogenous bases, so they are different. These differences also arise because, and for example, this thing, you can appreciate this anti and syn conformations of the nitrogenous bases. So what do they mean? I have the slide later that anti conformation actually means that your nitrogenous base is is not on the sugar, the plane of the sugar. It's outside the plane of the sugar. Whereas seen means your nitrogenous base is, is like within, is lying over the plane of the sugar. So these are two different conformations of the nitrogenous bases, right? So those are different between these different forms of DNA. Now, how do they arise? Why do different DNA conformations arise? That there can be various different factors. For example, it can be binding of proteins to nucleic acids, as well as it can be nucleic acid modifications. For example, methylation of adenine, methylation of cytosine, or deamination of several different nitrogenous bases, as well as that is DNA modifications. The not changing the nucleotide sequence, but changing the kind of chemical identity of the nitrogenous base, the chemical nature of the nitrogenous base. Not the chemical identity, but the chemical nature of the nitrogenous base. Yes. So Now, so what happens is, for example, this ZDNA, ZDNA is often found, as well as there are, among other factors, is the nucleotide sequence. The nucleotide sequence also plays an important role in determining conformations. For example, if there are repetitive nucleotide sequences, right? So, for example, polyadenine, polythionine. Then there is a formation of this. There is an increased propensity of formation of this triple helical DNA, where RNA DNA triple helices can be found, or the DNA DNA triple helices can be found via a very different kind of base pairing, which is non-Watson Crick, which is called Hoogsteen base pairing. Okay, and ZDNA is typically formed due to negative supercoiling, some torsional stresses which arise in the region ahead of the transcription fork, the transcription bubble, or also in many cases because of the DNA strand break or DNA damage. Right? Okay, so yes, so that's what that these are the conformations that I was trying to talk about. That anti is basically that conformation where the nitrogenous base is placed outside. Uh, that is, the nitrogenous base is not lying over the plane of the sugar molecule; it's lying outside. As seen means it is lying on the plane of the sugar molecule. Right. This DNA, how like any eukaryotic cell has such long DNA, but it's present in a very condensed manner within the nucleus of the eukaryotic cells. So that condensation happens because of the binding of DNA, because of the wrapping of DNA around DNA-associated proteins, which are called the histone proteins, right? And this mm, histone core, like consisting of four different histones, in total 8 histone molecules, and then the linker histone H1 together forms this nucleosome, and DNA is present in a very compacted and condensed manner within the nucleus of the eukaryotic cells. Now, this condensation states also change in the different stages of the stages of the cell cycle. For example, in the metaphase state, you get the most condensed state of the DNA, where you see the DNA condensed in the form of chromosome, right? Which are arranged on the metacentric plate. However, after that, DNA also gets decondensed, right? And thereby, this unwinding of DNA actually allows those DNA regions to get accessible to the transcription factors, to the replication machineries, which is necessary for DNA replication, as well as DNA transcription to form different kinds of RNAs. Okay. So, next, let's focus a bit on this on DNA replication, right? Which also have been studied in quite a bit details, have been taught in quite a bit details in the first week of this course. So DNA, as you already know, is a antiparallel double helix, right? So it is double-stranded, which is present in um, double-stranded, which are kept together um, via the non-covalent hydrogen bonding, the Watson-Crick base pairing between the purines and pyrimidines are two different strands. So for DNA replication, the first step is unwinding the two strands of the double helical DNA. So there is an enzyme called the DNA helicase, which unwinds the two strands of the double-stranded DNA. Now the helicase, after binding of the helicase, that is, if you try to imagine that the helicase binds to the double-stranded DNA, it opens the two strands of the double-stranded DNA. Then there will be a formation of a bubble, right, at the origin of replication, at a certain specific site of the double-stranded DNA, which is called the origin of replication, right? So after the binding of helicase, these two strands of the DNA are separated at the origin of replication, and a bubble is formed, which is called the replication bubble. Now, after that, the helicase keeps on moving in a certain direction. Okay. So in this case, the helicase is moving in this direction. This is the direction of the movement of the helicase. And you can imagine that there will be formation of, for example, if this is your DNA, right? Then this basically what is happening is, like initially after the binding of this helicase and separation of the double strand, there will be transcription bubble. Separation of the two double strand, there will be formation of the replication bubble. And then the this helicase enzyme will be moving, right? Will be moving in a certain direction. Also, one helicase can bind here and move in this direction, right? Mm, Now, so this becomes a replication port, right? This becomes your replication port. This is your replication bubble. This entire thing is a replication bubble, and this becomes a replication port. Okay. So now the helix. So in this figure, the helicase is moving in this direction, right? Now what happens after the single stranded DNA is formed? So the first thing is there is an enzyme called primase. Okay, so it binds to this single-stranded DNA and forms a small primer. Okay, forms a small primer. So now out of these two single DNA strands, one will be your leading strand, one will be your lagging strand. So what that really means is, for example, these are the two parent DNA strands, right? Which will be used as the templates for the DNA polymerase to add in new DNTPs and elongate and synthesize a new DNA strand, right? So, um, so for example, this so these are the template. These are your parent template DNA strands. That is the 3' direction. That is the 5' direction. And these RNAs are formed in the complementary manner, right? By the primase. So after the formation of the small oligonucleotides by the primase, so you can appreciate this fact that you have a double-stranded part and you have a single-stranded part. So this will be called, for example, here after just the formation of this small oligonucleotide by the primase, here will be the double strand and here will be the single strand, right? So that will be called the single strand double strand junction, where the DNA, which the DNA polymerase recognizes and where the DNA polymerase will bind, okay, on both this strand as well as on this strand. Now, so for this strand, now for both the strands, now what will happen is the DNA polymerase. There are different DNA polymerases, but this will be DNA polymerase 1. So this will be DNA polymerase C. So what it will do is it will now elongate a small RNA oligonucleotide in a certain direction. So if you remember the chemistry of polymerization of DNTPs that we discussed earlier, then you can appreciate this fact that new DNTPs can only be added to a 3' hydroxyl group of the already existing primer. Okay, so the DNA polymerase binds to this double strand single strand junction, and it allows new DNTPs to come in a DNA in a complementary fashion. That is, for example, if it finds one adenine next, then it allows a thymine to come in. If it finds a guanine next, it allows a cytosine to come in and polymerizes. And it polymerizes the incoming DNTPs by catalyzing the formation of the phosphodiester bond. And leading to DNA elongation in this direction. The direction of elongation is always from 5' to 3'. So 3' end you can see to be getting elongated. So if you look into this thing that for the lower strand, as the direction of polymerization of the DNTPs by the DNA polymerase will be in a direction which is opposite to the direction in which the replication path is proceeding. That is the direction of movement of the helicase, right? Now, so that is why this lower strand will be called the lagging strand, whereas the upper strand will be called the leading strand. Because for the upper strand, the direction of polymerization will be the same. The direction in which the DNA polymerase will be polymerizing the DNTPs will be the same in which the replication fork is moving, right? So now that is also a big problem that for this complex, how will it be possible to elongate this lower strand, for which the elongation needs to be done in the opposite direction of movement of the helicase or the movement of the replication fork. So actually, in real sense, in real life, how the problem is solved is that the lower strand or the lagging strand is actually looped. It's looped like this. Okay, this lower strand is actually looped like this, so that now you can see that the direction of elongation is this. Now, since because it's a loop, now the direction of elongation chemically remains the same. That is 5' to 3'. But because of the looping, what happens is that this complex consisting of the helicase and the DNA polymerase and several other proteins can move in the same direction of the direction of movement of the replication fork. But what happens is that because of this looping, the processivity of the DNA polymerase for polymerizing DNTPs of the lagging strand is much lower. What that means is processivity means how many number of nucleotides the DNA polymerase can polymerize at one go after binding the DNA or this double strand single strand hybrid junction and without leaving the DNA. So processivity and rate of polymerization are two different things. Processivity means how many nucleotides the DNA polymerase can polymerize after in one round of binding and release, like between one round of binding and release. And the rate of polymerization is how many bases the DNA polymerase can polymerize per unit time, right? Okay, so for the lagging strand, what happens is the processivity is much lower. That is, it can, for example, if for the, excuse me, if for the leading strand, it can polymerize 10,000 bases at one go. Then for the lagging strand, what happens is it can polymerize, for example, 50 bases at one go. That is, after polymerizing 50 bases, this strand is released from the DNA polymerase. And that is also necessary because the helicase is moving in this direction. So more of the single strand is being formed, which needs to be again replicated. So for the lagging strand, what happens is the synthesis of the new DNA strand happens in much more fragments or pieces, which are called the Okazaki fragments. And for each Okazaki fragment, the initiation of the DNA replication, that is initiation of elongation or formation of the new DNA strand, needs the formation of a new uh, oligonucleotide primer, which is found by the primase every time. And the DNA polymerase again binds and it actually elongates. It polymerizes 50, 100 bases. Then again, that is released from the DNA polymerase. Again, the new single strand is looped. The oligonucleotide is formed by the primase. The DNA polymerase binds and elongates. So that's how DNA replication happens. That's how DNA replication happens a bit differently for the leading strand and the lagging strand. Now, after formation of all of these Okazaki fragments, which consists of the RNA oligonucleotides and then the DNA 50 bases or 100 bases. So what happens is these, for both the leading strand and lagging strand, the RNA oligonucleotides, the primers basically, which are formed by the primase. So those are removed with deoxyribonucleose with DNTPs by DNA polymerase 1. And the gaps, for example, there will be some gaps between this, for example, RNA primer and the DNA strand. I mean, the gaps between the Okazaki fragments, most importantly, these gaps are actually joined together by ligases, okay, which leads to formation of continuous DNA nucleotide sequences in both the lagging strand and the leading strand, right? So although the lagging strand is formed in these pieces, in bits and pieces, kind of, in the form of the Okazaki fragments, it is not so that you can allow errors to happen in the lagging strand. So DNA replication is a high fidelity process. And these DNA polymerases, to ensure this high fidelity, also have their error correction mechanisms. Which is the 3' to 5' exonuclease activity. That is, DNA polymerase typically works in 5' to 3' direction. right? 5' to 3' direction, polymerizes DNTPs in this manner. But if it polymerizes, for example, one wrong DNTP, that is one mismatched DNTP, one non-complementary base pairing DNTP, then it also has this 3' to 5' exonuclear activity by which it can remove this mismatched wrong DNTP and again polymerize a new DNTP, which makes DNA replication a high fidelity process. That is, the error rate is very, very low. Okay? And however, errors also happen, which causes mutations and genetic diversities, and then also shape evolution and all. But yes, in general, DNA replication is a very high fidelity process for both the leading strand and the lagging strand. Okay, so let's go to the next slide. Yes, so now I'll also try to discuss some of the questions that you were asking, you have been asking in the discussion forum, right? So one question was that does only protein coding genes make proteins? So, okay, yes. So what I could understand from the question, what I could make, I am telling in that way. So I thought of introducing or discussing and explaining to you what are the coding regions and what are non-coding regions. What are coding regions of the DNA and non-coding regions of the DNA. So if you look at the definition of a gene, then a gene is that segment of the DNA that encodes for a gene product, okay, which can be RNA, it can be protein. So there are so many different forms of RNAs, mRNAs, rRNAs, tRNAs, like messenger RNAs, ribosomal RNAs, transfer RNAs, and so many other types of RNAs as well, like microRNAs, long non-coding RNAs, circular RNAs, etc. So among them, only mRNA is the RNA, which is again translated into the amino acids in the certain polymer of amino acids, having a certain amino acid sequence, which are called the proteins, which are the polypeptides that eventually fold into proteins. So genes are actually those DNA segments that code for a gene product, which can be RNA or protein. So that makes your coding regions, right? However, there are also, and for example, so there are several RNAs which also are not coding for protein, but still RNAs are also gene products and they have different functions of their own. For example, ribosomes are these ribosomal RNAs which perform several different catalytic activities and for ribozymes, right? And these tRNAs, they actually carry the amino acids into the ribosome machinery for polymerization of the amino acids in a certain sequence to form the polypeptides. When they carry these amino acids, they're called the amino acyl tRNAs. And various different other non-coding RNAs are also there, which play various different kinds of regulatory activities, like gene expression regulation, the genome organization or genome compaction within the nucleus, etc. However, so this forms your coding section of the DNA. However, on the non-coding section of the DNA also, there are non-coding sections of the DNA also. These are DNA regions or DNA nucleotide sequences which do not encode for any gene product, be it RNA or protein. Instead, these are DNA sequences which, however, they are not dispensable. They are also important. For example, these can be promoter regions where the transcription machinery binds. Right? This can be enhancers that lead to that ensure proper protein DNA interactions that also regulate gene expression and genome compaction, genome organization, and various important activities. So those are non-coding regions. However, those are also not dispensable. Those are also important. Okay, so yes, this was one question that what is the fate of the introns which are spliced out of the pre-mRNA to form the mature mRNA, right? So during transcription, for the production of mRNAs from the genes, that is the DNA, via transcription. So the genes consist of exons and introns. The introns can be thought of as a non-coding regions, which are typically spliced out of the pre-mRNA, and the exons are joined together to form the mature mRNA, which is then transported from the nucleus to the cytosol, where the ribosome machineries are present. They bind the mRNA and they translate the mature mRNA nucleotide sequence information into proteins in a simplified manner. However, in some cases, these some of these introns are also not spliced, and this lead to a diversity, a spectrum of different mature mRNAs, which are by a phenomena which is called alternative splicing. Anyways, let's not go into that complexity for now. So, to discuss this point, that what is the fate of the intron that is spliced out of the pre-mRNA? The primary fate is degradation of the introns. So what happens is during the splicing out of the intron, there is a formation of this lariat, like a circular RNA, which has a small tail. And the circularization happens because of the formation of a very unique phosphodiester bond, a 2' 5' phosphodiester bond. Now, in RNAs, you can, I think you can appreciate that they have ribose as the sugar or the monosaccharide, right? So they have 2' OH. So this 2' OH and 5' phosphate forms this 2' 5' phosphodiester bond that leads to the circularized intron with a small tail, with a small 5' tail. So, yeah. So the primary fate of these introns is that there are enzymes which cleave this 2' 5' phosphodiester bond, leading to the formation of a linear intron, which is then degraded via exoribonucleases, and um, the kind of nucleosides, the basically diphosphates, the diphosphates that are released, for example, adenosine diphosphate, guanosine diphosphate, cytosine diphosphate. Those will be again reused for DNA replication. Those will be again reused in the sense that they will be again phosphorylated from the deoxyribonucleotides, and then they will be again reused from the ribonucleotides, and then will be used for transcription. That is one fate, that the introns, the lariat introns, are debranched and into linear small RNA fragments, which are degraded by the exoribonucleases to again form the monomeric subunits that are again reused for transcription. The alternative, however, there are also some alternative fates. This intron lariats, in some cases, may be processed and modified into into stable stable RNAs, which are not readily degraded by these exoribonucleases. And this stability is primarily because of the processing and modification of those RNAs. And these small RNAs can actually perform various different important cellular functions, regulatory functions, like regulatory non-coding functions, like they can be small nucleolar RNAs, they can be microRNAs, circular RNAs, long non-coding RNAs, etc. They can regulate the gene expression, and they can perform various other functions. There was one question that would crystallization of proteins change the structure? So, and if indeed crystallization changes the structure of the proteins or the conformations of the proteins, how do we then the results that we get by techniques like X-ray diffraction and not accurate, then how to get the accurate results? So I will say that in short, the answer is yes, that crystallization of proteins typically people use different conditions, like different salt concentrations, and especially dehydration of a highly saturated protein concentration, highly saturated, high protein concentration solution. So, so that may lead to some three-dimensional protein. So these crystallization conditions can have an effect or impact on especially the three-dimensional arrangement of the secondary structures of the proteins. And this crystallization crystallize this the information, the conformational information obtained on crystallized crystal from their native or aqua solution conformations. However, I will like to point out that especially the secondary structures and the motifs will be quite similar. They are very less likely to be hampered a lot via these crystallization conditions, but the three-dimensional orientation of the secondary structures and motifs might be indeed hampered to a certain extent. And especially, anyways, crystallization, then these X-ray diffraction structures cannot give you the dynamic information of the fluctuations or the breathing of the proteins or changes of conformation depending on ligand binding, depending on different protein-protein interactions and all. That anyways, this crystal structure or X-ray obtained by X-ray diffraction will not be able to give you. The dynamic information it cannot give you. It's more of a static structure. And the three-dimensional structure might be a bit different from the native or aqueous solution protein conformations, which is caused due to the crystallization condition, salt concentration, dehydration, etc. So how to get the dynamic information and how to get accurate results to make conclusions and to, for example, develop drugs and to various other applications. So there, the answer lies in this fact that nowadays only the X-ray diffraction crystal structure of the proteins is not used for coming to conclusions. You actually couple it with several other informations about protein structures and conformations obtained via various different other techniques like nuclear magnetic resonance, cryo-electron microscopy, as well as high spatiotemporal resolution fluorescence microscopic information like single molecule fluorescence, Foster resonance energy transfer, and super resolution microscopic techniques like MinFlux. Okay, so these give you so many of the, for example, NMR will be in NMR can be done in solution. NMR can be done in aqueous conditions, and fluorescence experiments can readily be done in aqueous conditions. There's native conformations, and the cryo-EM also does not need crystallization, so it can also be a native conformation. And the high spatiotemporal resolution will also give you the spatial resolution as well as temporal resolution, which will also give you the dynamics of the change of the protein conformation. This we have already discussed about the DNA, different DNA conformations. Yes, now I will try to discuss some of the previous assignment problems so that it might be of a bit help to you in solving the week's assignment problems. So this question, they have asked that in the following figure, which pathway for the flow of information has not yet been discovered in eukaryotic cells? So not yet been discovered, that is to be stressed. Now you see that DNA is capable of, okay. So what are the options? Options is 7. 7 is, okay, 7 is this one. 7 means then DNA to RNA, which is transcription. 7 means this is 7. 7 is actually DNA to RNA, which is transcription, right? So that is discovered. 2 is RNA to RNA. That is also discovered. That is replication using the RNA-dependent RNA polymerase, right? So this is discovered. 7 is also discovered. It is transcription using the DNA-dependent RNA polymerases. 6 is saying DNA to protein. This is not discovered because DNA, there is no particular enzyme known which can read the DNA information from protein directly. You need transcription to happen to form this mRNA transcript. Its splicing happens to on the mature mRNA, then it moves to the cytosol where the ribosomes is necessary for translation. Okay. So 6 is not discovered yet. So that gives you the answer as out of these options. 4 is also discovered. 4 is also discovered because this is RNA to DNA, which is basically done by the class of which is done by the enzyme reverse transcriptase. This is RNA dependent DNA polymerase that is employed by a category of viruses, which are called the retroviruses, an example of which is the HIV. Right. Yeah. Okay. Now, the second question, the first accurate model of DNA was proposed by Maurice Wilkins and Rosalind Franklin had the data, which turned out to be very handy. Linus Pauling and Robert Cody were not, they proposed the triple helix, but the triple helix model, which was very different. All the nitrogenous bases were pointing outward, and the phosphoester bond kind of was was placed inside, with the nitrogenous bases pointing outside, which was not right. So here you can, I think, you can very well appreciate that the answer is Watson and Crick. Does DNA-dependent DNA polymerase make mistakes while replicating DNA? So yes, we have already discussed that this DNA replication, right? Where this DNA-dependent DNA polymerases come into picture. So that has a very high fidelity. That is a very low error rate. But yes, errors do happen. And because of these errors, there are mutations, and these mutations can often be deleterious, can often be disease-causing, disease-associated. However, these mutations in a very low frequency often turn out to be beneficial. And sometimes they end up increasing the stability of a protein, increasing the catalytic activities, the catalytic efficiencies of a protein. And then if you think it in the context of evolution, out of this genetic diversity that is created by these mutations, then that mutant is selected, which eventually gives the organism better or beneficial, advantageous phenotypes. So in short, the answer is yes. Indeed, the error rate happens, but the frequency is quite low. But it's not absolutely foolproof. Chargaff's rule for DNA states, so if you really know the Chargaff's rule, naturally, what it mentions is that the ratio of purines to pyrimidines is always 1:1 in DNA. Okay. Now that indirectly, so it is implied from this thing that, okay, if the ratio of purines to pyrimidines is always 1:1 in DNA, which is a double helix, then most probably is that a purine non-covalently hydrogen bonds or base pairs to a pyrimidine of the other strand. The inter-strand purine-pyrimidine hydrogen bonding keeps the two DNA strands together in the DNA double helix. But yes, so Chargaff never mentioned that adenine and thymine form double bonds and the guanine and cytosine form triple bonds. He mentioned the ratio of purines to pyrimidines is always 1:1 in DNA, from which it was implied that, okay, then the purine must be hydrogen bonding, or if DNA is indeed must be hydrogen bonding, must be kind of associating with the pyrimidine of DNA. Okay, what did the X-ray diffraction pattern of DNA produced by Rosalind Franklin suggest to Watson and Crick? Obviously, so that suggested the DNA has a double helix structure because of the missing fourth spot, right? So it suggested that DNA is a double helix. DNA is a double helix, right? Because of in the diffraction picture, there was a missing spot which indicated the double helical nature of DNA to Watson and Crick. Which of the following statements about DNA replication is not true, right? This you have to again be careful of, but not true. So Okazaki fragments are found on leading strand. That is true. DNA polymerase replicates DNA from the 5' to 3' direction only. That is very true, which we have already discussed. Okazaki fragments are formed on the lagging strand because in the lagging strand, the DNA polymerase has to the DNA is looped, right? Without looping, the DNA polymerase has to move in a direction which is opposite to the direction of movement of the helicase in the replication path. So to compensate for that problem, the DNA is looped, but that is lower processivity, and DNA polymerizes a certain number of bases, and then again it releases the DNA. It again binds the newly formed single-stranded DNA of the lagging strand. So the lagging strand, the DNA replication is discontinuous, right? The DNA replication is discontinuous, leading to the formation of Okazaki fragments. DNA helicase definitely unwinds and separates the two strands of DNA, and DNA helicase uses ATP energy, right? It's an ATPase. It uses the ATP energy to open or unwind this double-stranded DNA, which are held together by the complementary base pairing, right? So two DNA strands are joined by, in this question, the DNA replication is, so you can see that these three are true. And the fourth option, the DNA replication is continuous on both leading and lagging strand. That is false, right? Which is not true. So this will be your answer because DNA replication is continuous on the leading strand, but discontinuous on the lagging strand, right? So that gives you the answer. Two strands of DNA are joined by definitely non-covalent hydrogen bonds between the adenines and between the purines and pyrimidines of the two different DNA strands. Adenine double bonded with double hydrogen bonded with thymine, and guanine triple hydrogen bonded with cytosine. Okay. So the last three questions. So basically, which enzyme joins the Okazaki fragments on the lagging strand? So if I just mention the function of all these enzymes, then you will get the answer yourself. So DNA-dependent DNA polymerase, it polymerizes the incoming DNTPs in a complementary base pairing manner using the parent DNA as a template, right? So it doesn't join, however, the Okazaki fragments. Helicase unwinds and opens the DNA double helix into the single-stranded DNA. Primase polymerizes small RNA oligonucleotides for initiating the DNA polymerase binding and elongation. So DNA ligase is your answer, right? Which actually joins these Okazaki fragments on the lagging strand. If a single molecule of DNA, single double helix, undergoes 5 rounds of DNA replication, then how many molecules of DNA are created which do not have any original strand of DNA? This is a very interesting question. So to approach this question, you have to know this concept that DNA replication is semi-conservative. Okay. Now what does that mean? DNA replication is semi-conservative. What does that mean is, so for example, so you have initially a single DNA strand, okay? And these are your parental DNA strands, which I have marked in red. And what I will be doing is, I will be marking the new DNA strands that are generated using green, okay? So from this DNA, after one round of replication, okay? After one round of replication, if you look at the two double helical DNA molecules that will be formed, then you will see that, so for example, these two strands get separated first and the new strand is synthesized, right? Now, the thing is that you can see that there are two DNA molecules, right? And each of them has one red, that is the initial parental DNA. That's why DNA replication is actually called semi-conservative. That is half of, like, out of the two DNA molecules, each gets DNA is double helix, but only out of the two strands, one strand is a parental strand, right? So that's why it's semi-conservative. Now, if you keep on doing this, for example, if you keep on replicating these DNA molecules, then what you get to see, for example, from initially you had just one double helical DNA molecule. From that you get two DNA molecules. By DNA molecules, what I mean to say is double helical DNA molecule. From two you get four, right? So because it's a geometrical progression. Now, when you can think that when this DNA will be replicating, then this will be the template strand, one template, this will be the other template. So for the DNA where this green will be the template and the new green strand is formed, right? And same for that, where the green will be the template and the new green strand will be formed. So in this round, you can see that out of these four molecules, four DNA molecules, so two rounds of DNA replication has happened as of now. This is one round and this is one round. And this is DNA replication one, this is DNA replication two. So from one molecule, you have formed uh, two molecules. It's geometric progression. So after one round of replication, you have 2 to the power 1 number of molecules. After two rounds of replication, you have 2 to the power 2. That is 4 number of molecules. And out of these four molecules, you can see that two molecules don't have, right? Two molecules don't have any of the original strand of DNA. That is this red strand of DNA, right? So you can see that only if you see this pattern, then only two DNA molecules will be able to have this red strand of DNA. So in the next strand, in the next round, basically third round of replication, I'll have 8 DNA, right? So there also you can see that this stays constant. That only two molecules, that out of the 8, only two molecules have this single strand, have this initial parental DNA strand. So they have said 5 rounds of DNA replication, right? So after 5 rounds of DNA replication, the total number of DNA molecules that you will have is 2 to the power 5, right? Which means 32 number of DNA, 32 DNA molecules, 32 double-stranded, double helical DNA molecules. Out of them, only two will have this original strand of DNA. That means 30 molecules will not have, right? So that gives you the answer 30, right? And the answer is coming from this concept that the DNA replication is semi-conservative. That each of the daughter, after a single round of DNA replication, each of the two each of the two daughter newly synthesized double helical DNA molecules retains one um, out of the two strands, and one is the parental strand. So half, semi-conservative. If it would have been conservative, then what would have happened that the two parental strands would be staying together and two new strands will be staying together. Right? Which is not the case. So a nucleotide, now the last question, a nucleotide is composed of, now with that we have already discussed, that it consists of the sugar base, like deoxyribose sugar, to the C1 carbon is attached as a nitrogenous base, and to the C5 carbon is attached to your diphosphate, right? So the answer is phosphate, sugar, and base. Okay. So that brings to the end of uh, the discussion from my side. Now, you can ask me if you have any questions. I will be happy to answer. Okay, so if you don't have any questions, then we can end today and let's meet on the. Hope you will be able to do the week's assignments nicely, and let's meet on the next week's session on Sunday. Thanks.